Good afternoon, everyone, and welcome to the fourth session in the Introduction to Plant Pathology for Arborists webinar series. My name is John Gudritis, and I'm the Executive Director of the Texas Chapter ISA and your host for today's webinar. Now, we got a great session today, but before we get going, let's go over some housekeeping items so you'll know how you can participate in today's webinar. You're all familiar with this now, just go over it real quick. There's a screenshot of what you should be seeing in the upper right corner on your own computer's desktop. This is your control panel, and in it is your questions pane. Remember, if you want to ask a question, just type it in at any time, and Dr. Appel will answer as many as he can during the question and answer period. Welcome, everyone, to today's fourth session of the Introduction to Plant Pathology for Arborists webinar series. Our speaker today is once again Dr. David Appel, Professor Emeritus at the Department of Plant Pathology and Microbiology at Texas A&M University. Again, welcome, everyone, and let's get going. Dave, I'm going to switch it over to you.
Okay, John.
You're up.
Okay, I'm going to shut down the chat. All right. Good afternoon, everybody. As John said, this is our fourth session, the third session in the series on plant pathology for arborists. And I'm going to start with a brief outline just to tell you what's coming for today's session. We're going to talk about pathogen types today. We're going to go through each of the major types of pathogens that I often show on my screens when I give talks, but we don't go into a lot of details about each one of these. And as I think I mentioned before, plant pathology by some people is just considered to be applied microbiology. And this always shocks undergraduate students when we get into the plant pathology course and they find out that they're going to eventually need to know the difference between a bacteria and a virus and a nematode and a phytoplasma. And I'm going to try to introduce some of those basic concepts to you today, and using a lot of tree diseases as example. And then the next topic I'd like to cover, if we have time for it, is called the disease cycle. And I have shown disease cycles repeatedly down through the years in all of my discussions, and you will be familiar with the disease cycle, the generic disease cycle, when you see it. But I wanted to talk about it in a little bit different light today and emphasize why we consider the disease cycle to be so critical in plant pathology. As a matter of fact, an awful lot of our research in plant pathology is targeted at clarifying different parts of the disease cycle for a plant pathogen. So with that as just a brief introduction, let's go ahead and get started. Now, we finished off, I'm going to repeat a little bit here today, but I have modified the slides. And the first thing we're going to talk about are fungi. And this is the largest category of plant disease-causing organisms, not just with trees, but with all plants. And I've got a nice image of a typical fungal pathogen on the right here, Phomopsis canker on live oak. This is what we call a target canker. And you can see the reason why we call it a target canker, and it's because you can clearly see the interaction that occurs between a plant pathogen trying to cause disease and a host, in this case our live oak, is trying to inhibit the pathogen. And some people might say, well, the Phomopsis canker is winning the war over here on the right in the image. But at least the tree has been surviving for many, many years in the battle. And although someday a good blow is going to come along, and because of the girdling by the canker-causing organisms, the tree is going to blow over. But still, nonetheless, the fungus or the tree has been surviving year after year, reproducing, growing, and so it's kind of a stalemate. But let's talk about fungi for just a minute and talk about why they are the largest category of disease-causing organisms. First of all, they're able to produce specialized infection structures. And this is an attribute of fungi. Even though they are microorganisms, they have evolved the mechanism to produce a lot of specialized structures which help them grow in an ever-changing and oftentimes hostile environment. Some of those specialized structures are the infection structures, and we're going to talk about them in just a little bit. Also, they have specialized reproductive structures. We have many times talked about these. I mean, think fungal mat with oak wilt. That's a specialized reproductive structure produced by a fungus. And these specialized reproductive structures have evolved to maximize the ability for the pathogen to reproduce and distribute in the environment. Third, fungi are able to produce long-lasting survival structures so that they can withstand environmental extremes. We're going to talk about what some of those are in just a second. And another attribute of fungi is they are able to combine physical force. In other words, they can attach to the host and then produce hydrolytic enzymes. And what I mean by hydrolytic enzymes are pectinases and cellulases that break down the cell walls of the fungus, and combined with the physical force of the hyphae or mycelium, then they are able to force their way into the plant tissues. So in essence, they're making their own infection courts, whereas an awful lot of pathogens, as we're going to see, rely on some other entity to make the infection courts. The fungi can make their own infection courts. And then finally, this brings us back to the very first statement I made, the first bullet up there. They are able to overcome host defenses. In other words, they can produce specialized infection structures that interact with the host that will help them overcome host defenses. So we showed this slide before, and what I'm doing here is setting up the introduction to be able to go over this slide again, because I would like to point some things out on it again for just a moment. First of all, I have added the fact that we are looking at a necrotrophic pathogen here. A necrotrophic pathogen is a pathogen that when it attacks the host, the plant, it invades the host tissues and kills the host tissue. It tries to kill the host tissue. We're going to see this is in contrast to a biotrophic pathogen that when it attacks the host, it tries to trick the host into not realizing that it's being attacked so that the host tissues stay alive. We called this an obligate parasite in my original definitions, but plant pathologists like to think in terms of necrotrophic pathogens and biotrophic pathogens. And as I said, let's take a look at what a necrotrophic pathogen does here. And I hope you all can see my pointer. You're going to notice that there are these little red arrows and yellow stars and blue dots and red squares, green dots, and black dots. You're going to see some of these are depicted on the outside, and these are products being produced by the fungus in order to gain entrance into the host tissues. For example, they might be the hydrolytic enzymes that I mentioned. They could be proteinaceous enzymes, in other words, enzymes that break down proteins. But nonetheless, we've got some of these things, and the term plant pathologists use for this are effectors. And we've got some of them on the outside of the plant cell, the green dots and the red dots. And we can see that they're being produced as the fungus tries to invade the plant cell. Now, some of these, like the red dots, are going to be enzymes that break down cell walls and plasma membranes. These are real disruptors. This is a weapon that the fungus has, some of these fungi have, in order to kill plant cells and then eat the contents of the cell that they've killed. But notice down here, we've got one of the apoplastic. It means it's being produced outside the cell wall. One of them has been picked up by a plant receptor up here in the right-hand corner. You can see that the plant has now detected that the pathogen is there. Now, this may be some kind of secondary metabolite. It may be a product that the fungus doesn't even use to in pathogenesis. But our our host here does have the ability to detect the pathogen. And if you look right on the inside of the cell, it says signaling cascade initiating plant cell defense. So now the plant cell knows that it is being picked, that the fungus is out there and trying to gain entrance. And all of a sudden, it's going to start producing all kinds of RNA molecules to tell the nucleus over here that we're in trouble and we got to kick into a defensive mode, which at this point the host cell is going to start producing different kinds of compounds that will be antifungal, natural compounds that the host has. It will be antifungal, maybe break down the fungal cell walls with chitinases. Remember when we talked about fungi on last Thursday, we talked about the fact that chitin is in the cell walls of true fungi. And this is enough to dwell on this slide. What I'm trying to do here is to illustrate where research has taken us in the past 20, 30 years, as far as our understanding of how this war goes on between the fungus and the host cell. So let's review this necrotrophic fungal pathogens again. Necrotrophic fungal pathogens obtain food from dead organic matter or dying cells of living plants. Necrotrophic fungal pathogens try to inhibit the defense mechanisms the plant is going to have. Like I said, chitinases, proteinases that the fungus has. And also, the fungus is trying to kill the plant cell itself, because if the plant cell detects that it is being attacked, it will go through what we call programmed cell death, which produces a whole lot of obnoxious compounds that are toxic to the fungus. And the fungus is trying to stop that from happening. So it's going to kill the host tissues very rapidly. These are such diseases as leaf spots, blights, root rots, cankers, some of the vascular wilts. These are considered to be typical of what a necrotrophic fungal pathogen can do. We talked about the facultative parasites and the facultative saprophytes. Remember, you just put the term in there, sometimes parasite and sometimes saprophyte, for these necrotrophic fungal pathogens. But they have degrees of parasitism and degrees of being a saprophyte. On the right, we have the biotrophic fungal pathogens. These are fungal pathogens again. They obtain their nutrients only from living plant cells. We call them obligate parasites in some of the other terminology. They usually kill their hosts very slowly. They do not want the host to die. As I said, they're trying to trick the host. They don't want the recognition that this is a fungus trying to break through the cell wall and grow through into the plasma membrane and start having an exchange of nutrients. Usually biotrophic fungal pathogens have very narrow host ranges, as opposed to the necrotrophic fungal pathogens, which usually have broader host ranges. These are such things as nematodes, phytoplasmas, viruses, the downy mildews. We don't have a lot of downy mildew pathogens on trees that we talk about, a big problem in the vineyards, and also the powdery mildews, which are obligate parasites, are biotrophic. So just as a reminder now, we've got all these interactions going on, and every one of these attributes that we see here, the effectors, the plant receptors, all of these are conferred by the genetics of the fungus and the genetics of the host, so that we end up having genes for resistance. We talked about this on Thursday. We've got these genes for resistance in the host cells that tell the host to turn something on, And we've also got the effector genes in the fungus, which are constantly turning on different genes as well for these different colored, in our case, these little cartoons, the stars and the arrows and that sort of thing. Each one of those is genetically controlled. And so therefore we have this setup between the host and the fungus, that we have the genomes of the two organisms are in this battle. They're signaling back and forward to each other. They're trying to turn off and on each other's genes. They're trying to influence each other. We call this in plant pathology the gene for gene concept. And if you look at this first bullet, for each gene that converts virulence, the effectors, to the pathogen, there is a corresponding gene, a receptor, in the host that confers resistance to the host, and vice versa. That should be not just versa, but vice versa. Generally, we know what these genes are in a lot of host-parasite interactions. This research has been worked out for many, many, many crop diseases, so that the breeders can then determine which genes for resistance they need to work into the genetic background of a hybrid that they're trying to make in order to increase the resistance to fungal pathogens in the crop. And as I said, we know the details about this. We know about the dominance of the resistance genes and the genes for susceptibility, the lack of resistance in the host. We also know about the genes for virulence, the ability to infect in the pathogen, and the genes what we call avirulence, inability to infect in the pathogen. And I've always thought it was very interesting that one of the first people back in the 60s that began to see this happening in trees was Al Shigo with the CODIP model, because all this fits together with how the compartmentalization of decay in trees model works as far as hosts go. We don't know much about what the genes are, however. Don't get me wrong. The research that has been done on trees is very, very limited. And this brings me back to when I talked about oak wilt and the resistance responses to the oak wilt organism. Observations with inoculation studies in the bur oak that we talked about showed that there were portions of the vascular system that when inoculated, the vascular system turns dark and is full of all these gums and gels and dark compounds accumulated where the tree was inoculated. And these are resistance mechanisms. The trouble is we just don't know what the genes are. The work has not been worked out in the genome of the oak tree to learn about these resistance genes. Now that it looks like some laboratories are going to start working on this, but we're just not there yet. Tyloses, another thing. We know that in this inoculated red oak down here, that the tree, the red oak, even a red oak, this is an extremely susceptible tree. Even a red oak has some things that we consider to be resistance mechanisms, like the tyloses here that turned on in a tree, a red oak, a pin oak, that was inoculated with the fungus. So the tree produced tyloses. It's just that it didn't produce them quickly enough to slow down the spread of the pathogen. Now that gives us some clues about how tyloses fit into the pictures. Tyloses are a generalized, non-specific defense response in the vascular system to lots of different things, not just specifically to the oak wilt fungus. And therefore, if you look at a tree on the right here, there were no tyloses that were detected in the vascular system of this tree. But the problem is that the red oaks just don't detect the fungus quickly enough. So they're missing one of these receptors that I talked about. And that's where the breeders someday, 10, 15, 20 years from now, when people finally start working on oak wilt resistant breeding, that's where they're going to have to focus is why are the red oaks not recognizing the presence of the fungus, whereas the bur oaks do seem to recognize it. Okay, some examples of biotic fungal pathogens, necrotrophs and biotrophs would be powdery mildew. I already said that it's a biotroph. Leaf curl would be a necrotroph. Black spot on elm is a necrotroph. Rust on post oak, however, is a biotroph. These are called rust diseases. A lot of you have noticed this disease on the lower portion of post oaks some years. Some years we have it, some years we don't. And this rust disease is what we consider to be a biotroph. Fire blight, necrotroph, virus, is a biotroph. So I hope you're getting an idea here that that all pathogens are not equal. They all are very, very different in the way that they cause disease, and as such, they are very different in the way that they're controlled. Same thing with vascular problems. However, let's take oak wilt, for example. Oak wilt is a necrotroph, but if you recall, the many times I've said it, the oak wilt fungus is a very, very good parasite, but it's a very, very poor saprophyte. So therefore, it's a facultative saprophyte. It's just sometimes it's a saprophyte. It behaves very much like a biotrophic organism in a lot of the stages of its disease cycle. Laurel wilt, not so much. I would call it a pure necrotroph. Bacterial leaf scorch, again, starts edging towards a biotrophic type organism. It's extremely difficult to grow in the absence of the living host. So therefore, I would probably tend to push it towards a biotroph, even though most bacteria are necrotrophic. Dutch elm disease right here, very easy to culture in the laboratory, and we consider it to be a really good necrotroph. We've got another vascular wilt in Texas that we don't talk about a whole lot, Fusarium wilda mimosa. I don't even have a good picture of this organism. I diagnosed it a few times when I first got to Texas, back when I was looking around for things to work on. Well, that didn't last long. And I had a little more time that I began to have once I really got caught up in oak wilt. But we do have Fusarium wilt, classic vascular parasite that is a necrotroph. Okay, let's move on to bacteria and talk about them a little bit. Now, bacteria are what we call prokaryotes. They do not have their nuclear material organized in a true nucleus like the eukaryotic fungi do. So therefore, prokaryotes are a little bit at a disadvantage. Their DNA is organized as circular chromosomes in the cell. And if you look in the upper left-hand corner, you can see what the chromosomal organization of the DNA in a bacterial cell doesn't look like a typical nucleus. So therefore, their nuclear organization is different, and therefore they aren't as sophisticated as the fungi. They don't have nuclear membranes. They don't have mitochondria in the classical sense. So therefore, they're very limited in being able to produce specialized structures like the fungi do. Now, we have to recognize that bacteria are actually a smaller category of disease-causing organisms in plants, even though in animals and man, there are a lot of bacterial diseases. So we've got a difference going on here in pathology as far as the disease-causing organisms go. Typical symptoms by bacteria are leaf spots, scorching, chlorosis, cankers, blights, wilts. Notice these are the kinds of diseases that necrotrophs, the necrotrophic types of fungal pathogens produce, and most of our bacteria are necrotrophs. These are unicellular. They can't produce specialized structures like the fungi can. Now, you do have some fungal organisms that produce chains, but still we don't see this habit a lot in plant pathogens. we do see some serious bacteria. Don't get me wrong here. In the fruits, especially stone fruit trees, there are some extremely serious bacterial problems. But oddly enough, we don't have as many bacterial problems in the forest trees. And so therefore, we're not going to spend an enormous times on bacteria, but I do want to talk about the fact that they are very effective plant pathogens. These organisms cannot make their own infection courts. They rely on natural openings and wounds made by some other agent to be able to infect. Natural openings would be such things as lenticels, stomata, hydrothodes. This is how bacteria get into a plant. The one advantage they have is they have huge reproductive rates. Bacteria have what we call binary fission. In other words, one bacteria divides into two bacteria, two into four, four into six, six into eight, or two into four, four into eight, eight into 16, 32, 64. And this is a remarkably rapid way for an organism to reproduce. And the bacteria are the absolutely supreme organisms on Earth. They undergo this binary fission very rapidly. So you think about a bacteria that gets drawn into the stomata of a plant leaf, and it's in a susceptible plant, and it realizes it is in a good spot because it's absorbing compounds out of its environment, and it begins dividing. Pretty soon, the stomate is plugged up with bacteria, and they're oozing out of the stomate onto the surface of the leaf. The reason they are really good at colonizing host tissues is because they produce hydrolytic enzymes. Now, fungi produce hydrolytic enzymes as well. I've already talked about it, pectinases, cellulases, but the bacteria are really good at it. They produce enormous amounts of hydrolytic enzymes. They are also very good at producing host-specific toxins. Toxins, these are small compounds, small-sized proteins that are extremely lethal to plant tissues. And they spread very easily by wind, splashing rain. They spread on seeds. Now that isn't important to us arborists and natural resource managers dealing with trees, but in the crop world, this is very critical. Bacteria spread very effectively on seeds. And I'm going to just talk about how bacteria cause diseases for just a second, and then we're going to move on off of the bacteria. If you look at the left here, though, we've got a bacteria called Pseudomonas, and we've got a picture of it. It causes bacterial speck on some host right there. Not sure what the host is. Don't even care a whole lot because we're looking at the molecular level right here. And it's got what we call a secretion system that it inserts its effectors into the host cell. Look at that. Y'all are plant pathologists now. You know what I mean by effectors. And then these effectors then begin to do their work on the plant cell. And keep in mind that some of the effectors that the bacteria is inserting into the cell are hydrolytic enzymes. So these things really make a mess of their host cells. We've got crown gall right here is a very effective pathogen. It's got what we call a type 4 secretion system. The effector that it inserts into the plant cell is a piece of its genetic material. And when it does that, it then the genetic material is incorporated into the host cell, and then it tells the host cell to start dividing so that the plant starts making the gall that we see. So this is a very different kind of pathogenic system going on. On the right, we've got black-legged potato. Now this is the one I wanted to show. Type 2 secretion systems that are cell wall degrading enzymes. These are the rotting organisms. Let's say, for example, you put a peach in the crisper in your refrigerator, and you leave it there for a long period of time. And you come back and you pick it up, and it's just a mass of liquid inside the skin of the fruit. You know, your fingers poke through the skin and that sort of thing. It's because these kinds of organisms like Erwinia produce enormous amounts of cell wall degrading enzymes. This happens a whole lot with I'm trying to think of the like cabbage, or maybe can't, it's completely lost my train of thought on it. However, but when you have vegetables in your crisper and they turn to a mush, a mass, cilantro is the word I'm thinking of. You keep cilantro in a plastic bag and you come back 3 months later, I'm sure none of the rest of you do this like I do, and it's just a green liquid. These are the types of bacteria with type 3 secretion systems. And then finally, we do have vascular wilts with bacteria as well that live in the vascular system. These are a whole different kind of group of organisms. And I just wanted to make sure that they got in there to let you know that some bacteria do produce vascular wilts in plants. As far as trees go, we're familiar with some of these organisms. I just put them up here to remind you. Indeed, we have slime flux right here is produced by a bacteria. We've got bacterial leaf scorch down here in the lower left right here caused by Xylella. We do have crown gall right here on the right. I'm sure you all are familiar with that. And then in the upper right-hand corner, we've got fire blight. All of these are Erwinia, are types of bacteria that produce tree pathogens in the manners that I described. Now, I do want to talk about phytoplasmas. I bring these up a lot on my list of pathogens, and we are going to just briefly mention them because we don't have a lot of serious problems with phytoplasmas in Texas. There is a problem in the northeastern United States that we suspect that we have here in Texas. It's called ash yellows. A colleague of mine, the original diagnostician at the Texas Plant Disease Diagnostic Laboratory, Dr. Larry Barnes, he started the TPDDL, as a matter of fact, back in the early 80s, just as I got to Texas A&M, and he did fool around and was pretty sure, using the tools we had back in the early 80s, that he successfully detected ash yellows in Arizona ash. They were Arizona ash that were that had died back, and they had these kinds of sprouts at the base of the tree and along the lower trunk. He was able to use the tools at the time to confirm ash yellows, which is a phytoplasma, but nobody's ever done any work since with it. Poor Arizona ash just doesn't get the attention that it needs. And then we've got Wong Wong Bing down here, citrus greening. I've talked about that a little bit with some of you in some of my talks. It's caused by a yellows type phloem limited bacteria. Now these are bacteria, but they don't have cell walls, so they're what we call pleomorphic. In other words, they don't have a typical shape, where whatever they're contained in, they will kind of mold to the shape of however they're crowded into the plant tissues. These are all obligate parasites. They're transmitted by leaf hoppers that feed on the phloem of the tree. We get chlorotic leaves, witch's brooms, poorly developed root fruits like we do with HLB. So even though we do have phytoplasmas in Texas, tree people really don't worry about them a whole lot. I'll bet you we're going to find that there are more phytoplasmas in the future as soon as a forest pathologist comes along that has the time and money to work on it. These things cause disease. There are a lot of them, 300 known hosts. There are a lot of leaf hoppers, plant hoppers, psyllids. that feed over here in this cartoon right here at the top. They feed into the phloem. They pick the phytoplasmas up, and the phytoplasmas. This is called a propagative persistent vector, and let's say it's a psyllid, and it's because the phytoplasmas actually live and multiply inside the vector. And so therefore the infectious insect is very effective because these things are circulating within the vector. And now they are infected, but they don't cause a reduction in the fitness of the vector, but they do actually condition the vector to go around looking for the hosts if they wanted to. Fascinating research. Wish we could get into it more, but we really don't have it right now, and that's as much as I want to say about phytoplasmas. Viruses, I've said many times, we don't have a lot of virus diseases, but nonetheless, I don't want to ignore any of these categories because plant pathologists are expected to know a whole lot about each one of these, whether you work on it or not. So we usually end up have short courses during our graduate studies on every category of pathogens, even though we go on in careers and only study one or two of them. Viruses rely completely on wounds for penetration. Usually these are mechanical wounds, but they may also have insect vectors just like the phytoplasmas. Most plant viruses consist of a single strand of RNA, not DNA, like COVID and a lot of the human viruses. Plant viruses are mostly RNA. There are a few DNA viruses, and I've recently had to work with one in the vineyards. They're very much easier to work with than the RNA viruses. Usually designated by the disease they cause. So let's say, for example, tobacco mosaic virus causes a disease in tobacco. It causes a mosaic-like symptom, and that's how they get their names. But one of the curious things about viruses is tobacco mosaic virus actually causes a real problem in tomatoes. So it's a lot like cotton root rot causing problems in elm trees, in Chinese elm trees. The first name that the pathogen gets is the one that sticks. They are absolute obligate parasites. They can only multiply in living cells. They're tiny little things. Again, tree fruits do have a lot of serious virus problems, but when you get over into forest and shade trees, they drop off. They have been implicated in forest tree declines, particularly in Europe, but there's not a lot of proof of what they do. Let me see. I thought I had another slide. I thought I had a picture of island disease. If any of you, island disease of hackberry, it's a kind of a mosaic disease of hackberry. And anywhere you live that there's a lot of hackberry, if you look at it, you will be able to see chlorotic island virus of hackberry. I thought I had a slide of it in there, but we're moving on to nematodes. Nematodes are unsegmented roundworms. They are not real worms. They're classified uniquely from the worms because they have some attributes actually of insects. Most of them are soil inhabiting. They're very tiny. You can see them with your naked eye. I can't anymore, but if you have a suspension of them in a test tube, you can kind of see them. But nematodes have all the internal organs that human beings do: muscles, digestive systems, nerves, you name it. They're curious little roundworms. They are mostly soil inhabiting. Some of them get into the vascular systems of trees, and there is recently one that has just been found affecting forest trees. So let's talk about a few of these specifically. Now here's pictures of them. Let me back up. I just want to make the point that if you look in the upper left-hand corner, all of parasitic nematodes have what we call stylet. That's very much the same kind of structure that an aphid has that feeds on leaves. It's a little spear that the organism shoves into a plant cell and then draws the nutrients out. So that if we look, and you can't really see it on the pine wilt nematode right here, on the feeding end, rather as opposed to the rear end of the nematode on the feeding end, it's very hard to see, but there will be a stylet up there that the nematode uses to feed. Pine wilt nematode is actually very common in Texas. It does get on lots of different pines, but the most susceptible host we have is Japanese black pine. And that's because Japanese black pine, we found out very early on, was very susceptible. And we introduced, the pine wood nematode is native to the United States, and it was introduced into Japan and caused a huge problem on this native pine species in Japan. But we do see the pine wood nematode on loblolly pines over in East Texas sometimes. These in Japanese black pine, it does cause branch dieback. The first time I ever saw it was in a bunch of Japanese black pine that were planted by the mall when I first got here in College Station, and those trees were planted just about the time I got here. They survived about 5 years, and they were all killed by the pine wood nematode, Monocamus species. It is vectored by what we call pine sawyer beetles. The new disease caused by viruses, which is a big problem now, it's spreading out of Ohio into the northeastern United States. It's very curious. It's called beech leaf disease. The symptoms of beech leaf disease are down here in the lower right-hand corner, and they are very peculiar. You can look up through the crown of a tree with the sunlight in the background and see these patterns of chlorosis and very dark green leaf patterns. This thing was just discovered and described in 2012. They have no idea where it came from. It got introduced into Ohio somehow. They're still doing research on the nematode itself. It is spreading rapidly on beech. This is a big problem. This is one of those things that is a reminder to all of us first responders out there. Always keep on the lookout for new things going out there in the woods. You never know when you're going to find something brand new. The name of this nematode is Lidolinchus crenacea. It's just restricted to beech right now, so we don't have to worry about it. I don't know of anybody that's planting a lot of beech here in Texas. We do have one curious disease, though, on pecan called the root knot nematode, Meloidogina incognita. This is the granddaddy of all plant parasitic nematodes. Huge problem on cotton in Texas. Very, very serious cotton disease. As a matter of fact, a lot of breeding efforts have gone on to incorporate the resistance to this particular nematode into the cotton that's grown in Texas, particularly up on the high plains. But we do have a species that does attack pecan, hickory, and walnut. Now, this is a root parasite. Now, we're not going to go into the disease cycles of the root knot nematode. It just take too long. However, I do want to talk about the fact that you may actually encounter this on pecans. If you are called out to look at a pecan and you've got a dieback, and if you're on sandy soils, now that's important. Root knot nematodes love sandy soils. If you're on high clay soils, then this is not going to pertain. But these little worms are able to swim around in the sand. Huge problem on golf courses, as a matter of fact. When they install all that sand, they set up the conditions for nematodes and all. And that's why golf course managers hate nematodes. But if you are able to get at the root system, if you look at this image in the lower left-hand corner here, the images, you can see the knots. They are very prominent on the root systems. So therefore, this is a very good diagnostic tool. If you're dealing with a pecan in sandy river bottom, someplace like that, You can get at the root system because you've got an unknown dieback. I just want to put it in there that it could be a problem. And I'm going to tell you, 41 years as a tree pathologist at Texas A&M, and this is the first time I've ever mentioned the root knot nematodes on pecans. So aren't you glad you came today? Look at that. Let's talk about parasitic higher plants now. Uh, uh, these are true flowering plants. Now, they do have modifications. They don't have true root systems. If you look down here in the lower right-hand corner, instead of a root system, they have what we call cortical strands. That this is the germinating seed of the parasite, and in this case, it's a mistletoe. Doesn't have to be a mistletoe. There are others out there. But when the germinating seed penetrates the branch, it will grow down and it will grow these cortical strands that produce sinkers that hook up with the vascular system of the tree. Now, sometimes these organisms only hook up with the xylem, and therefore they only draw water and inorganic nutrients out of their host. Other parasitic higher plants will hook up with the phloem as well as the xylem. And therefore, these are more serious plant parasites because they actually draw contents out of the phloem as well. And I hope that you can all see why there would be an important difference between the two types. We've also got, and I say that right up here, some require only water and inorganic nutrients. Others lack chlorophyll, and therefore they're yellow, and they require sugars. And that's what we consider the difference between hemiparasites and holoparasites in the world of parasitic higher plants. Hemiparasites are only partly parasites because they only need water and inorganic nutrients. Holoparasites are completely parasitic on the plant and are usually yellow. They have no chlorophyll. And we've also got root pathogens and stem pathogens, and I've already talked about the modified root system, so I'm not going to talk about that. But let's talk about the two most common parasitic higher plants on trees. And we have both of these in, well, no, actually three. We have all three of these, sorry about that, in Texas. One of them are dodder, and you will see this out in the hill country. And John Gudritis is very familiar with dodder because we worked on an invasive species of dodder once that invaded trees. And notice how it's yellow. It's a stringy vine. Notice it has no chlorophyll. It's usually orange. This is a native dodder. This is the invasive species that got into Houston. We call it giant Asian dodder. However, out in the hill country, every once in a while, you're going to see in live oak a native tree dodder too. A little bit more down in the southern range, although I used to run into it all the time out at Fort Hood, in parts of Fort Hood when I was doing research out there. The other true parasite that I want to mention is dwarf mistletoe. Now, this is a yellow plant. Notice how it's real tiny right here. This only occurs on conifers out in far west Texas. So if you ever get out the Big Bend and you decide to take a long hike up to the south rim, you're going to be able to run into conifers out there that have this little stubby plant. It's yellow. It has no leaves. It will produce flowers though. Remember now, these are mistletoes. A very curious little plant. Believe it or not, this is one of the most serious forest tree diseases in the western United States, is what we call dwarf mistletoe. Now, you know what? I left out a slide of leafy. Oh, no, here's the leafy mistletoe. Sorry, I forgot to mention leafy mistletoe. Notice leafy mistletoe is green. Y'all are familiar with this one. It does produce seeds and flowers, that sort of thing. And notice that it is green, so therefore it is only a hemiparasite. It only draws water and inorganic nutrients out of its host. parasitic higher plants in some parts of the world are some of the most serious of all plant parasites, particularly in Africa. There are many parts of Africa that have root parasites of sorghum and corn and some of their other crop plants that are really bad. But we're not going to talk about those today. We're going to finish off with the abiotic pathogens. We've been over this already. I'm not going to go through it in great detail. Certainly in Texas, I've already talked about how I call them pathogens. I'm not going to go through this again. Just bring that hot button issue up again. But I do just want to remind you that freeze damage is a big problem here in Texas all of a sudden. And we know a lot about how freeze damage damages trees. One of the big issues that we have to be aware of is the fact that trees are able to acclimate and prepare themselves for freeze damage, but it has to be a healthy tree because it takes metabolic energy for a tree to acclimate. So therefore, if you've got a weakened tree that doesn't have the storage carbohydrates that it needs, then therefore it's not going to acclimate as well, and therefore it's going to be more susceptible to freeze damage. We know that acclimation is actually a lot like dehydration in a plant. It's controlled dehydration, so to speak. Water is drawn out of the plant cells so that the solute concentrations of the plant cells get higher, and the water is moved out into what we call the apoplast. We've been over these, the spaces in between the cell walls and the xylem of the plant, of the tree, and therefore the tree moves the water out of the plant cells so that those living cells will not freeze and get ice crystals in there and get disrupted, whereas the water is then out in the apoplast. That's the dead portions of the tree, the non-living portions of the tree, where some freezing isn't going to be bad. True, we could still get damage if the tree drops down to extremely low temperatures like we did have in 2021, but it's all about acclimation in trees. Acclimation is what determines whether we get short-term freeze damage or long-term freeze damage in trees. And I'm running short on time, and I just want to get through the two types of injury. We have short-term, which we can see right away as soon as we get into the growing season, and then we have the long-term, the heart rots, and the predisposition to wood bores and that sort of thing that occur years and years and years after the freeze damage occurs. I'm not going to go through drought. We've been through it a whole lot, but it is an abiotic pathogen as well, just to remind you where we are here, and then herbicide damage. And I'm going to stop now because we're going to start up with the disease cycle on Thursday, and then we're going to get into plant disease management from a plant pathologist's perspective. We're going to be talking about, I'm going to focus on biological control because that's a hot issue right now in trees, and also olive plant pathology. A lot of research going on in biological control, and then a little more about host resistance and breeding for resistance. And with that, I'm going to go ahead and stop.
Okay, thank you, Dr. Appel. Gene, are you there? Gene Gehring is our Texas Chapter ISA Assistant Director, and he's going to give your audience questions to you, Dr. Appel. As a reminder for all you out there, you can still submit questions to the questions pane in your attendee control panel. Take it away, Gene.
All right. The first question up is, does fire blight and cotton root rot affect cottonwood trees?
Fire blight does not, and cotton root rot does affect cottonwood. It's just a coincidence, but yes, cottonwood trees are susceptible to Phytotoxicopsis omnivora, but not fire blight. Did I say that right, Gene?
Yes.
Okay.
I'm not going to disagree with what you just said.
Yeah. Okay. I started getting the word cotton and cotton root rot and everything mixed up. Okay.
Can you evaluate the claim that archaea or archaea sprinkled on the ground in oak wilt centers can, well, it says guarantee cure of oak wilt, but impact oak wilt?
There is no biological reason that I know of why archaea would be effective in controlling oak wilt. Now the people who put archaea out there, and archaea are very, very difficult organisms to work with and that sort of thing. But nonetheless, the people that put them out there as a commercial application, I know that they claim that they turn on certain genes in the plant, and we have seen that certain bacteria like Agrobacterium does have the mechanism to turn on genes in the plant. And we know that other bacteria can do that. So I am not going to dispute that there may be something going on there. However, in the systems that we do know where it occurs, because we do know the genetics of how certain bacteria affect plants, and they may turn on a little bit of systemic acquired resistance, it's called in plants, but it would not be enough to affect cotton root rot, let's make a point there, or oak wilt or Dutch elm disease or any of those kinds of diseases. It may be that a mixture of archaea makes a plant a little more vigorous because you are replenishing the microbial population of the soil, but archaea get their name for a reason, and I would be very leery of trying to get some archaea going in a very, very, very complex microbial population of soil. It's not going to be an easy fix. And we'll talk more about that when we get to biological control.
All right. The next question is, are crown galls synonymous with burls?
We do call the symptom of Agrobacterium crown gall burls. However, there are a lot of tree species that produce what I would call natural burls for genetic reasons. Those are often the ones that woodworkers try to get a hold of. If you saw that picture of the gall that I was standing next to, that had a lot of rot into it. Now, there are some wood turners that like to use those galls that have the rot in it because they produce a lot of character in the bowls and flasks and that sort of thing that they make. However, they are very hard to work with because they're full of decay. And that's usually what happens with these old crown galls called by Agrobacterium. They get all full of decay, very difficult to work with. So a lot of times if you go to these lumber mills that deal in specialty woods, they'll have some native crown galls there for you to work with, but they're also going to have burls from tropical tree species that they have gone out and purchased and brought in where the wood is a little more solid, and you can get the beautiful effects of the healthy wood that these tropical species produce.
All right. And then the last question on this list is there any chance that the complete slide set from these excellent presentations might be made into a booklet?
Well, Gene, that's a question for you and the organizers. I'm not averse to it. I'm pretty sure I've done a good job of giving credit to all of the slides that I've snatched off the internet. But that's up to ISA. I'm going to leave that up to you guys. But I would certainly work with you on that project, Gene, if we all decided to go for it.
What we might be able to do, Dr. Appel, is take each one of your webinars is going to be on demand on our website, and we'll include in each one of those the presentations that goes along with it. Would that be okay?
That's okay with me.
We'll put it in a PDF and include it with the on-demand webinar. Well,
You know,
I think that's it on the questions, but before we close,
John,
sorry.
could I get a comment in real quick? This is the first time we've ever done this before. And on Friday or on Thursday, you're going to be asked, I assume, to fill out a questionnaire. And I am really interested in feedback. We would really like to know what your opinion is and suggestions if we ever do it again. Thank you.
All right. And that does answer the last question that came up. It says, if not a booklet, would the slides be available for the class attendees? And
Okay.
yeah, I mean, that will be up there. And when you were talking about little leaf mistletoe, when we were in Yellowstone on a hike, our guide must have thought I was crazy because I held everybody up while I was taking pictures of
Right.
mistletoe.
It really is a terrible pathogen in the Rocky Mountain commercial forestry. It's really bad.
All right, John.
Great. Okay. Well, before we close, let's talk CEUs. Everyone who gave us a TDA pesticide applicators number and or an ISA certification number at registration will have a certificate emailed to them after the final webinar this Thursday. The certificate will list all your CEUs and you will get credit for all the sessions that you attend. As a reminder, you cannot get TDA CEUs unless you attend the live online sessions. Session 5 is this Thursday, as Dr. Appel mentioned. Again, look for your session links 1 day and 1 hour before the start of the session. I just mentioned that all of the sessions, as we get them edited, will be put back up onto our website under webinars. So the first 3 are already up there. This one will be up probably tomorrow or the next day whenever we get it edited and the questions. So go there if you want to review it, and if you should have already gotten the master code, the discount code for the webinar, because if you attend this, you don't have to pay, of course, to go back in and watch it. So in closing, thank you, Dr. Appel, and thanks to all of you for attending today's webinar, and thanks again for taking care of the Texas trees out there. Stay safe out there, and we'll see you on Thursday.
Thanks, everybody.
Thank you.